Hello, and thank you for joining us. My name is Rose Chaffee-Cohen, and I am a curriculum specialist with Mini PCR Bio. I'm here today to walk you through a great DNA and electrophoresis lab called Inherited Cancer Risk Genetic Testing for Lynch Syndrome, which we launched in the fall of 2024. First, let's start with some introductions. I mentioned that I'm part of the Mini PCR Bio team. And our goal is to make biotechnology more accessible to everyone, especially teachers and students. Our equipment is durable, portable, and most importantly, affordable, which makes it useful for a number of different applications. So our end users include scientific researchers who are working at remote field stations, citizen scientists who are identifying locally foraged mushrooms, and of course, teachers who are introducing essential biotechnology tools to their students. Our curriculum team is composed of a group of scientists and educators who are passionate about making biotechnology education accessible for teachers. As I mentioned, my name is Rose. I'm pictured in the bottom left, and I taught high school science for 16 years, including biology and bioethics. And I know firsthand how putting real biotech in the hands of your students has the power to inspire them and engage them and build confidence. At Mini PCR, we offer innovative lab activities as well to give students that hands-on experience with biotechnology so that teachers can show them how the tools are used to solve real-world problems. And our Inherited Cancer Risk Lab presents one such scenario, which introduces genetic testing for an inherited disease risk. Now, genetic testing is something most students are going to encounter in their lifetime, making it very relevant. And not only that, but disease risk factors are often misunderstood. So this lab provides an opportunity to teach about those inherited risk factors. In addition, the lab provides an opportunity for students to apply their understanding of pedigrees to track inheritance and make testing recommendations. And all of this is explored through an engaging case study of a family who's navigating testing decisions and results. So all of these factors make this a great lab for a general biology course, AP biology, or a health sciences course. So we ask students to imagine themselves as a doctor who is treating Ariel, a 30-year-old who is recently diagnosed with cancer. Now, because of her age, she's relatively young, and a family history of cancer, Ariel received genetic testing, and this identified an inherited mutation that's associated with increased cancer risk. So today, we're going to test DNA samples from 3 of her family members to see if they carry that same inherited mutation. So just to give you an outline of what we're going to do today, we will load the samples now so that we can analyze the data in real time before the end of this webinar. And then we'll go back and review the lab scenario while those samples are running. We'll also practice interpretation of the gel before reviewing and interpreting our actual results. And we'll end with implementation information and some free educational resources from Mini PCR Bio. So right now, I'm going to switch over to my bench cam to show you the lab kit, the prep, and the sample loading. So when you purchase this lab kit, you're going to receive a bag with all of the samples in the mail, and you'll see that you have the instructions for storage. This particular lab you can store in the freezer. And where I start is I go to the store page for the lab, and under curriculum downloads, I download the instructor guide. That's going to give you really clear instructions for both how to set up the timing for this lab, all of the teacher prep, and just all of the information about the lab overall. And the prep for this lab is really easy. You can do a lot of it in advance. So up to 1 week in advance, I like to aliquot out the samples into little tubes for my students, just to make sure everything's ready for them. They don't need to share the larger vials. And so you can see here we've got 4 samples that we're going to test along with a ladder. And so I give one of those to each group. The other thing you can prepare in advance are the gels. And again, this is super easy. I made our gels today. I made a 2% gel using sea green tabs. These are all-in-one agarose tabs that have the agarose, the sea green stain, and the buffer. So you just dissolve about 20 mils of water for a 2% gel. Make sure that's dissolved. Put it in the microwave until it's clear and it boils. And then you can pour that solution into one of these casting trays. And so for this lab, I used a 6 well comb, but the blue gel system also comes with 9 and 11 wells. But for my students who are beginners, I really like to start with those larger wells as they're learning how to load their samples. It just gives them a bigger target. So I poured my agarose here. I'm going to just remove the comb. And then I have a tray that can go right into my blue gel for loading. So it's really easy to prep those in advance. You can store them for up to 5 days in an airtight container in the dark. Don't need to add anything else. And then they're ready to go. So that's what I've done in advance. And when you're doing that gel prep, it's also a great time to make your buffer. And so if you order buffer from us, we often send it in a powdered form where you just add 600 mils of water, dissolve that, and then you're ready to go. So today I'm going to run our samples in our blue gel system. This is our most popular system because you can both run the samples and visualize them all in one. But you can run this in another electrophoresis system that runs the samples horizontally. It has to have fluorescence visualization. So this includes our Bandit STEM electrophoresis kit as long as you add on the View It illumination kit, and this makes it it's a really affordable option for running these samples. The other piece of equipment you need is a micropipette that can dispense 10 microliters. And I really like our fixed volume mini pets. This one's 10 microliters because they have the same basic mechanics as a traditional adjustable pipette. You get the first and second stop, the tip ejector. And so that allows students to learn how to micropipette, but it's a much lower price point. And students aren't going to break this by overwinding the dial like is a common mistake with adjustable pipettes. So with that, we are ready to start loading our samples. I'm going to zoom in and give you a better view of the blue gel here. So I'm going to take off my lid. I already have my gel in there with buffer, just covering it. You can see that. And so again, we're going to load a ladder first, and then our 4 samples. We're going to load 10 microliters into each well. So we'll start with our ladder. And when I'm teaching students how to pipette, I usually do that before they've actually done a lab. First, I remind them, new tip every time, just to avoid cross-contamination. And I also remind them to steady themselves. So right now you might not be able to see, but I am holding my wrist. And then the third thing is to remind them they don't need to press the tip of their pipette all the way down to the bottom of that well. If they just wiggle it right in that opening, I know it's going to go a little fuzzy for a moment while it focuses. If they just wiggle it there, it's the samples are going to sink because they're denser than the buffer around them. And that way they can avoid puncturing the bottom of their well and losing their sample. And lastly, I like to encourage them to really pipette slowly. If they push the sample out very quickly, they might get a big plume of sample that ends up leaving the well. And so by really practicing that slow, patient pipetting, they're more likely to get their samples loaded correctly and to not have any errors related to interpreting the gel later because their samples weren't loaded correctly. So with that, I'm going to zoom out a little bit just to show you we've got our samples loaded. All we need to do is put on the blue gel lid, and we're going to press this power button. You'll notice that'll stay green. So that means electricity is flowing through the entire system. And when we're ready to visualize this later, we'll use the illumination button here to get that blue light. One of the things my students always loved to do was to take a time lapse during when the gel was running. And an easy way to do this is if you use our Folda View, now it comes in this great silicone version, you can pop that right on there and put your camera here and set up that time lapse, and you'll get a really great video that students love to share. And what's better than having them talk about a lab that they did in class? So with that, we're going to switch back over to our slides and talk about what's in these samples. All right, we're going to let our samples run. This only takes about 20 minutes. And while that's happening, I'll share how you can set your students up for interpreting these genetic testing results. Just like each of our labs, we provide background information that walks students through the biology concepts that they're applying in the lab scenario. Today, I'll be providing you with a teacher-focused version of that background, but if you want to review the full version, you can find it in the student guide for the lab. Now remember, the focus of this lab is understanding genetic testing for inherited cancer risk, and we're using Lynch syndrome as an example. First, it's important for students to know that cancer is caused by mutations that lead to uncontrolled cell division. Now in the student version of the background section, we look more closely at the difference between somatic mutations and mutations in reproductive cells. And it's this difference that leads us to our next point. Some people, because of their genetics, have a higher likelihood of developing cancer. Now, an estimated 5 to 10% of cancer are associated with these inherited mutations. And these are genetic risk factors that increase the chance of developing cancer in their lifetime. Again, today we're focusing on a specific type of inherited cancer risk called Lynch syndrome. Lynch syndrome is caused by mutations in mismatch repair genes. And these genes encode mismatch repair proteins that repair the mistakes that are made when DNA is copied. So if a mutation occurs, functional mismatch repair proteins are going to repair that mistake. But without functional mismatch repair proteins, a mutation that occurs when DNA is copied is going to remain in the DNA, and that mutation then becomes a permanent part of the cell's DNA. So someone who has dysfunctional mismatch repair proteins are going to accumulate a greater number of mutations over their lifetime. And this greatly increases the risk of them developing cancer because cancer is caused by the accumulation of mutations. Later on in the webinar, I'll also share a lab extension activity that dives into how certain mutations are associated with cancer specifically. So Lynch syndrome is diagnosed using genetic testing that identifies these mutations in mismatch repair genes. And even if an individual inherits one copy of a mutation in a mismatch repair gene, that does greatly increase the risk of them developing cancer, and that is enough to diagnose them with Lynch syndrome. Lynch syndrome is associated with an especially high risk of developing colorectal cancer or endometrial cancer before the age of 50. This is often how it's first diagnosed. There may be a pattern of early onset cancers of this type in one's family. Individuals that are diagnosed with Lynch syndrome are encouraged to start routine cancer screening earlier than is typically recommended. And it's early detection that really can improve treatment outcomes for these individuals. Relatives of somebody who have been diagnosed with Lynch syndrome, they may also wish to be tested to see if they inherited the same mutation. And if they carry that same mutation, they're also going to have an increased cancer risk. So remember, we're imagining ourselves as a doctor who's treating Ariel, who was recently diagnosed with colon cancer and is already undergoing treatment. Now, because she's relatively young, remember she's only 30, and she has a family history of cancer, she received genetic testing. In this lab, we also provide students with pedigrees for Ariel's family, and these track the incidence of cancer. And with a basic understanding of genetic inheritance, students can analyze this pedigree here, and they'll likely notice that Ariel has a parent, a grandparent, and an aunt who died of cancer before the age of 50. Remember, that's considered early for cancer. So this family history is one of the reasons why she was recommended for genetic testing. So Ariel's genetic test revealed that she carries an inherited mismatch repair gene mutation, and her mutation consisted of extra DNA inserted into the MSH2 gene. This is just one example that is specific to this scenario, but another family with a history of Lynch syndrome may have a different mismatch repair gene mutation. So since we have diagnosed Ariel with Lynch syndrome, there are several immediate family members who are also undergoing genetic testing for the same MSH2 mutation. So today we are testing DNA samples from Bethany, Ariel's sister, Caleb, Ariel's uncle, and Devorah, Ariel's aunt. Now, this lab focuses on the results of a genetic test. So the procedure you're completing with your class is gel electrophoresis, which is what we started at the beginning of this webinar. But electrophoresis is actually the last step of a longer experimental workflow. So I'm going to share what comes first. First, DNA would be extracted from a biological sample like blood. And we have to remember humans have a lot of DNA. But we're really just interested in zeroing in on one small segment of DNA, like the MSH2 gene. So this is where PCR, or polymerase chain reaction, comes into play. PCR allows scientists to make billions of copies of just the DNA that they want to study. In our case, this is a portion of the MSH2 gene. So once we have copied the MSH2 DNA, we can use gel electrophoresis to determine whether the same MSH2 mutation that Ariel inherited is present in our other samples. So steps 1 and 2 were already done for us, and you saw me start step 3, the gel electrophoresis, at the start of the webinar. At that time, I told you the samples represented DNA, but now you know that what we're examining is billions of copies of just the MSH2 DNA segment that were generated using PCR. I also want to provide you with a little bit of background on gel electrophoresis. Gels are commonly made using agarose, which is a sugar that comes from seaweed. And at the microscopic level, which you're seeing on the left-hand side of your screen, the agarose forms a web which acts like a size filter, where some molecules can move through this web more easily than others. We also position a negative and positive electrode on either side of this gel. So when we apply an electric field, the DNA, which is also a negatively charged molecule, is going to move through that web-like gel towards the positive electrode. And during that process, those smaller pieces of DNA, which have an easier time moving through the web, are going to travel farther in the same amount of time, Well, those longer pieces of DNA are going to get caught up in that web and they're not going to travel as far. So this is what that looks like to our naked eye when we're looking at a real gel. We load the samples into wells at the top of our gel. And in our samples, there might be a mixture of DNA molecules of different sizes and lengths. But as the gel runs, the DNA molecules are going to separate based on length, with the longest pieces of DNA traveling the shortest distance. And the shortest pieces are going to travel farthest, right? The shorter ones are smaller, they can move through more easily. We'll also load a DNA ladder into lane 1, and this consists of DNA pieces of known length. So this allows us to compare our sample bands and estimate their size in base pairs. So before we examine our results, we're going to practice the same analysis students will do as they work through the lab guide. All of the following questions are included in that learning lab guide, and they can be used with students just to ensure that they're ready to analyze the real results. So keep in mind, the MSH2 mutation that Ariel carries is an insertion with extra DNA. So with our visual, the functional version is represented by a 600 base pair PCR product, whereas Ariel's MSH2 gene mutation is represented by a 1200 base pair PCR product. And gel electrophoresis is going to allow us to separate and visualize the MSH2 DNA from our patients so that we can determine if they carry that same mutation that Ariel carries. So first question, we already know that Ariel carries one copy of the mutated MSH2 gene. So what are her gel results going to look like? Right? Our schematic of the chromosomes shows she's got one copy of each type of MSH2 DNA. And so we're going to see two different bands, one for the 1200 base pair segment and one for the 600 base pair segment. Well, what about somebody who has two copies of the functional MSH2 gene? Each of those copies would be 600 base pairs. So we're going to end up with a single band at 600 base pairs. This is sometimes a question that confuses my students because they imagine, but we have two copies of this gene. But remember, they're both the same length here, so they all travel together, making a single band at 600 base pairs. I'm going to switch back over to our bench cam so that we can see what our results look like in the blue gel at this point. And what we'll need to do is turn on our illumination. You notice I've dimmed the lights in my room just to take out some extra ambient light, making it easier to see these results. And if I'm ready to start my interpretation, I like to turn off the power button just to keep the samples from running further down the gel. If we leave that on, they'll continue to run all the way off the gel eventually. You may prefer to keep the samples running longer than we have. This is about a 20-minute run because that ladder will continue to separate further. But at this point, we can definitely interpret our results because we have some good separation in our sample bands. I'm going to zoom in a little bit so that you can better see all of those lanes. Excuse the dog hair, that's hard to avoid in my house. But we've got our ladder in the leftmost lane, and then we have samples for Ariel, Bethany, Caleb, and Devora in lanes 2 through 4. And so we will be able to interpret those in a moment. I also just wanted to point out, you might notice below those, you've got some sort of bluish hazy bands. Those are just the loading dye from our samples, which move much faster than the DNA bands themselves. They are not useful for interpretation. They're just kind of a remnant that's left behind. So we can ignore those completely. So let's talk about what these results represent. Okay, so we have seen our real-time results in our blue gel, which are also represented here on our slide. This is a 20-minute run of the results for this lab. but let's take a look at each one step by step. So we might first ask, who carries the same MSH2 mutation that Ariel has? And in this case, Ariel is almost acting like our control because she has both a band for the 1200 base pair mutated MSH2 gene and a 600 base pair band that represents the functional MSH2 gene. So Bethany also carries that same pattern in her bands, one of each. Caleb has a single band that is 600 base pairs long, and that represents the functional MSH2 gene. So he has 2 copies of that band. And lastly, Devora, again, has one of each band, a 1200 base pair segment and a 600 base pair segment. That leads us to who is going to be diagnosed with Lynch syndrome. And remember, it's enough to just inherit one copy of the mutated MSH2 gene to be diagnosed. So Bethany has a positive Lynch syndrome diagnosis, Caleb is unaffected, and Devora would also be diagnosed with Lynch syndrome. So after students run the lab protocol and interpret their results, the lab guide also challenges them with critical thinking questions and claim evidence reasoning prompts. And I like to remind students that these results represent diagnoses for individuals and for a family. And so we ask them to reflect on what the next steps may be for each individual. We know that Ariel is already undergoing colon cancer treatment. Bethany, Ariel's sister, it may be recommended for early cancer screening for colon cancer and endometrial cancer every year, since now we've identified that they're at heightened risk for developing cancer. Since Bethany is also relatively young, this information might inform their family planning decisions. Caleb tested negative, so his cancer risk is not increased due to his MSH2 gene. However, being that he's 48, he's already at the age where colon screening every 5 years is the standard recommendation. So he should follow those guidelines. Devora is also Lynch positive. She's a bit older, and we've identified that she has an increased cancer risk. So she too will be recommended for more frequent cancer screening. We also provide students with a more extended pedigree for Ariel's family, where they can demonstrate an understanding of inheritance and who's at risk for inheriting this MSH2 mutation as they make some additional testing recommendations. Interpreting this pedigree requires only a basic understanding of inheritance, primarily that offspring inherit one copy of the MSH2 gene from each biological parent. So if we look at individual M, who is Micah, both of their siblings inherited the MSH2 mutation, presumably from their father, who died of colon cancer at an early age. So there's a chance that Micah may have inherited it as well, though it's really not certain, and they should get tested to confirm this. A similar rationale can be applied to individual I, who's Alana. I would also recommend them for testing. Given that they're older than 50, I might predict that they're Lynch negative, but we really can't be sure unless we do genetic testing. Now, since Devora was Lynch positive, it's possible for her to pass this mutation on to her daughter Fern, who I'd also recommend for testing. However, remember, Caleb tested negative, so he can't pass it on to his child Harbor. So in that case, testing wouldn't be necessary. And even with the testing recommendations that we would make, there are reasons why individuals decline to have genetic testing performed. This is something that we also explore in a bioethics role play extension activity. You can also get the answers to all of these questions by emailing answers@minipcr.com. Just email us from your school email account, and please make a note of your school and position there, as we do check that you're actually a teacher asking for the answers. So we have just walked through the core of the student lab guide. But this lab also includes some robust extensions that allow you to add even more depth to the activity. In our extension from Lynch syndrome to cancer, we walk through how Ariel, who started out with a genetic risk factor, eventually did develop cancer due to mutated mismatch repair proteins. This led to an increased mutation rate and critically driver mutations. In this lab extension, we provide some review questions that help you check for student understanding of these concepts. And I think one of the highlights of this lab is our extension that serves as a bioethics role-play activity. The role-play itself introduces Devora's daughter, Fern, who is almost 18 and is considering whether to get genetic testing when she turns 18 or not. She's planning to meet with a genetic counselor and is weighing the input of her parents as well as she makes this serious decision. And this extension was developed to provide the instructor with lots of guidance, scaffolded background, and practice to develop students' skills in ethical decision making more broadly. This extension culminates then in that role play activity. So this is a great lab if you're looking to introduce genetic testing or inherited disease risk. Remember, it allows students to practice tracking inheritance through pedigrees, and we have some great extensions to explore the mechanisms of cancer development and bioethics. If you're interested in buying the kit, it's $67, and that includes DNA samples for 8 groups. The equipment that you'll need is a horizontal gel electrophoresis system that has fluorescence visualization. And you'll also need a micropipette that can dispense 10 microliters. The teacher prep is super easy. You're just making gels and dispensing samples for your groups, and you can complete the core of this lab in only 45 minutes. Our lab store has a lot of resources that you can use in your classroom, including the teacher guide and student guide that you can download. We also have e-worksheets, which are editable, downloadable Google Docs that you can assign directly in your LMS, and PowerPoint slides with great visuals that you can download and edit to fit your classroom needs. We've also made alignment easy for you by sharing how our labs align with Next Gen Science Standards, AP Bio, TEKS, BASE, and Common Core ELA standards. And you can also find all of this on the store page. And feel free to reach out if you have alignment questions. Lastly, if you're looking for additional lab activities that explore topics in human genetics, disease, or health sciences, Mini PCR Bio has several other options. First, our sickle cell genetics lab also explores a genetic testing scenario, this time involving sickle cell disease, which allows your students to review family member profiles and predict results using Punnett squares, which they can confirm then with gel electrophoresis. The Viral Diagnostics Lab challenges students to diagnose patients with either seasonal influenza or an emerging virus using gel electrophoresis. This lab's a great introduction to viruses and also includes an extension on using genetic data to track viruses, where students examine viral sequences and analyze phylogenetic trees. Lastly, if you want a lab that includes PCR, I would recommend our cystic fibrosis lab called Missy Baker Missing. In this lab, students connect genotype and phenotype as they better understand the genetic disease itself, and they use it to solve a missing persons case. So I hope we've given you plenty of food for thought today and provided you with the tools to try out this lab. Thank you so much for joining us for this webinar, and we hope to see you at a future workshop or conference.